An obvious characteristic of many terrestrial plants is that they have green leaves. Woody plants must also be very rigid. In order to account for these characteristics, we must examine the material which makes up plants and animals. All plants and animals are made up of cells. Most cells are so small we have to use a microscope in order to see them. Cells are like little boxes or compartments which contain living structures. They are capable of dividing in order that plants and animals may grow. It might be difficult to imagine that trees of this size are made up of microscopic boxes or cells. The cells which make up the leaves show many characteristics common to all plants. We can identify these characteristics by using a microscope to examine a transverse section of a leaf. We can see cells in different layers. This section was prepared by making a vertical cut through the leaf. These cells are called palisade mesophyll cells and appear long and rectangular. In 3 dimensions, the cell appears cylindrical with rounded ends. When the leaf was sectioned, the palisade cells were cut vertically down their long axis to give a longitudinal section. The 2-dimensional shape we see down the microscope is rectangular. If we look under the microscope at the longitudinal section of the palisade cell, the main plant cell structures can be identified. The cell wall is a rigid box-like structure which gives the cell strength and encloses the fluid of the cell. The cell cytoplasm bathes the cell components or organelles and contains chemicals important in the functioning of the cell. Plant cell cytoplasm is rich in granules of starch. The chloroplast is an organelle which contains a green pigment called chlorophyll. The chloroplast is responsible for photosynthesis, and the chlorophyll gives the leaves their green colour. Chloroplasts are found in large numbers in the palisade cells. The nucleus of the cell contains a very important biological chemical called DNA. The nucleus is responsible for coordinating the chemical activities in the cell, such as the release of enzymes, and it is important in cell division. The vacuole of the cell contains a watery fluid known as cell sap. The vacuole gives the cell support and prevents it from wilting. Now we can complete the diagram of the cell by adding the vacuole and the nucleus. If we make a horizontal or transverse section of a palisade cell, the 2-dimensional shape we see down the microscope is circular. Under the microscope, the transverse section of the palisade mesophyll cell looks like this. Again, the cell wall, cytoplasm, chloroplasts, nucleus, and vacuole can be identified. The irregular shape of some of the palisade cells in this section is due to the fact that they are not always cut through the middle. There is a layer of cells above the palisade mesophyll, which is known as the upper epidermis. This cell layer forms a continuous outer covering over the leaf. Likewise, there is a lower epidermis covering the underside of the leaf. You may be asked to carry out an investigation which involves staining onion epidermis cells with iodine. portion of epidermis can be removed from the leaves which make up the onion bulb. The epidermis is a sheet of cells 1 cell thick. It is transferred to the iodine stain. A cover slip is placed over the preparation. The slide is blotted to remove any excess stain, and the epidermis can then be examined using the microscope. Under the microscope, we can see the epidermis cells linked together to form a continuous sheet. The cells are regular in shape as they have not been sectioned. As the cells are transparent, we can identify some of the structures we saw in the palisade cells. Here we can see the cell walls of 2 adjacent cells. The black dots are granules of starch which are found in the fluid cytoplasm of the cell. They have been stained black by the iodine. The cell nucleus is visible in all the epidermis cells, and so is the cell vacuole. Epidermis cells do not have chloroplasts as they do not carry out photosynthesis. These cells, taken from inside the human cheek, have been stained with methylene blue, so they appear blue in colour. Under high magnification, it is noticeable that these cells have no cell wall. All animal cells are enclosed within a flexible membrane, which allows the cells to change shape more easily than plant cells with their rigid walls. The cell membrane is also present in plant cells, but it is not usually visible as it is pressed against the cell wall. The cytoplasm of animal and plant cells has a variety of functions. Most animal cell cytoplasm is rich in glycogen granules. Glycogen is the animal energy storage product, equivalent to starch in plant cell cytoplasm. In most cells, the nucleus is clearly visible. The animal cell nucleus has the same basic functions as the plant cell nucleus. It contains DNA, it is responsible for coordinating chemical activities, and is important in cell division. Human red blood cells are produced in a tissue called bone marrow and are broken down in the liver. Once in the bloodstream, they do not need to divide and so have no nucleus. Human cheek cells have small organelles called mitochondria, which are common in many animal cells. These structures, seen here using an electron microscope, are responsible for the process of cell respiration. They break down food to produce energy. You may wish to discuss and compare its function with that of the chloroplast in plant cells. Human cheek cells do not have clearly visible vacuoles. However, some animal cells do have vacuoles. These single-celled animals live in water and excrete water from their cytoplasm by the use of a vacuole. You may wish to discuss why animal cells differ in function to plant cells and so have contrasting structures.